Dear students, today we will talk about histology of the organs of male reproductive system. So, male reproductive system includes two groups of the organs. There are inner and outer organs of male reproductive system. Inner organs, they include testes. Testis is the main reproductive gland. Seminiferous ducts, which carry spermatozoa from testis to outer environment, and also there are accessory glands, seminal vesicles, prostate gland, and bulbourethral glands, which produce different fluids and help in the movement of spermatozoa. So today we'll talk mainly about inner organs of male reproductive system and their histological structure. And there are two groups of the functions of organs of male reproductive system. There are reproductive and endocrine functions. Reproductive function is the main function and it includes production of spermatozoa or spermatozoons, male gametes, which are necessary for the fertilization, for the reproduction. And also, male reproductive system provides endocrine function. It's production of male sex hormone, which is called testosterone. There is a group of androgens, and male main hormone is known as testosterone. And both functions are provided by testes. So they are mixed glands which provide reproductive function, production of spermatozoa, and endocrine function, production of hormones and or other organs they help in the providing these functions and they are accessory organs of male reproductive system. So how they are related? The main gland of male reproductive system is testis, and the main function of testis is production of spermatozoa, and they are needed, they are necessary for the reproduction. And after spermatogenesis, it's a process of production of formation of spermatozoa. Those sperms, they are going to the seminiferous tract, special system of tubules, which carry spermatozoa from testes to outer environment, which is needed to the fertilization. And also there are special accessory glands, and they include seminal vesicles. Here we can see them. It's prostate gland, and also there are bulbourethral glands, and they produce special fluid, different kinds of fluid, which is released in the lumen of seminiferous tract. Here we can see fluid and it's released in the seminiferous tract. So spermatozoa which were released by the testes and they are released in the seminiferous tract, they are mixed with fluid which contains different nutrients for spermatozoa, also water which is needed for the movement of spermatozoa. It makes an environment for their movement. So spermatozoa plus fluid, and this mix is known as semen. So semen after the movement through the seminiferous tract is released to outer environment. So its main principle of the functioning and relationships of the organs of male reproductive system. And we'll begin from the main glands of male reproductive system. They are testes. So here we can see section and outer view of testis and its anatomical pictures. And let's talk about histology and microanatomy of testis. Here we can see that outer and the innermost layer which covers testis, it's called tunica vaginalis testis. It's continuation of the peritoneum. Here we can see process of descent of testis, and peritoneum forms processus vaginalis which covers testis. So it's part of the peritoneum and it has visceral and parietal layers, and between them there is a cavity. So testes, they are located in scrotum, and there are numerous layers which cover the testes, and the innermost, the closest layer, the closest structure to the testis, it's tunica vaginalis testis. And here we can see that testis is covered with tunica vaginalis. It's continuation of peritoneum, so it's covered with mesothelium, simple squamous epithelium, and thin layer of loose connective tissue. It's tunica vaginalis. But testes, they have own layers, so we will remove tunica vaginalis, and here we can see only own layers of testis, and we can see that testis is covered with connective tissue capsule. Here we can see dense connective tissue which covers testis, and it's called tunica albuginea. Its own capsule of the testis. Here we can see it. It's tunica albuginea. And also there is an mediastinum testis. Testis, it has bean-like shape, and here in this invagination there is a mediastinum testis, special region, anatomical region. And here we can see that between the tunica albuginea and mediastinum testis there are connections, and those projections of the dense fibrous connective tissue which connect capsule and mediastinum, they are called septa. So here we can see capsule, septa, these layers of connective tissue, and its mediastinum, and there are structures which form the skeleton of the testis, so-called skeleton. It's dense fibrous connective tissue which maintains the shape of the testis and protects another more soft tissues. So here we can see tissues and structures which are formed by dense fibrous connective tissue. And also there are special regions which are formed by loose fibrous connective tissue. They are softer and they are called lobules. So septa divide the testis into 200 to 2.5 of lobules. Here we can see lobules, and they have pyramidal or triangular shape, and they are separated from each other by the septa. And basis of the lobule, it's loose fibrous connective tissue with blood vessels, with nerves, but there are some structures which are located inside the lobules and which provide the main functions of the testis. So loose connective tissue of the lobules, it's called interstitium. It contains Leydig cells which produce testosterone, and essential structures of the lobule are seminiferous tubules, from 1 to 4 per lobule. Here we can see interstitium or stroma of the lobule. It's loose fibrous connective tissue, and inside this connective tissue there are seminiferous tubules, special convoluted tubules which produce spermatozoa. It's the place where spermatozoa are produced. And here we can see seminiferous tubules, and they are located in each septa, in each lobule. So here we can see localization of seminiferous tubules, and you should know that each lobule contains from 1 to 4 seminiferous tubules. And tubules, they have epithelial lining. They aren't formed completely from connective tissue, so they are in other structures. They have lumen, they have wall, and they provide main functions of the testis. They produce spermatozoa. They are seminiferous tubules. And also there are special tubules of seminiferous tract which carry spermatozoa from seminiferous tubules. There are straight seminiferous tubules or seminiferous tubules recta, and it's rete testis, efferent ductals, and it's ductus epididymitis, and they form special part of testis which is called parenchyma. So testis, they are gland-like organ or mixed gland which has reproductive function, or it may be described as a kind of exocrine function. It's production of spermatozoa, if we consider spermatozoa as an exocrine product. So it's a special part which provides reproductive function. It's convoluted seminiferous tubules. They form parenchyma, and also there are endocrine cells which produce hormones. That's why it's mixed gland or organ with endocrine and non-endocrine function. And also there is stroma. It's a non-specific part of testis which provides, which maintains parenchyma and helps to provide its functions. So here we can see stroma and parenchyma of testis. Stroma, it's capsule, it's interlobular septa and mediastinum. It's loose and dense fibrous connective tissue and also interstitium of lobules. It's stroma, it's non-specific connective tissue which contains blood vessels, nerves, and it helps to provide functions for parenchyma. And parenchyma is maintained by stroma. It has different tubules, it has different specific structures which provide specific functions of the testis. So testis is a parenchymal organ. It has parenchyma and stroma. Stroma is non-specific and supporting part, and parenchyma is specific part of the organ which provides specific functions. But stroma, parenchyma cannot exist without stroma. Stroma is a filler of the testis. It maintains structures of the testis and helps to provide functions of the testis. So, to make a conclusion, testis, it has stroma and parenchyma. Stroma is non-specific part of testis. It it includes dense fibrous connective tissue which forms capsule, septa, and mediastinum, and also there is loose fibrous connective tissue which forms intralobular interstitium, forms the basis of the lobules. And also there is parenchyma, which includes seminiferous tubules, convoluted seminiferous tubules, and seminiferous tract. Tubules, they produce spermatozoa, and seminiferous tract provides movement of spermatozoa. And parenchyma is specific part of the organ, which provides main function of the organ. And testis may be described as an organ with endocrine and non-endocrine function. Endocrine function is production of hormones, which is provided by the special cells of the connective tissue, and non-endocrine function, which is main, its reproductive function, its production of spermatozoa. And in some cases, testis may be described as a mixed gland if we consider production of spermatozoa as an exocrine function. So let's go to parenchyma of testis, to the seminiferous tubules, convoluted seminiferous tubules, and to the process of formation of spermatozoa, which is called spermatogenesis. Here we can see in other pictures which show the structure of testis. Here we can see connective tissue capsule or tunica albuginea, special name for capsule in the testis, septa, which divide testis into the lobules, and each lobule contains from 1 to 4 convoluted seminiferous tubules. Here we can see capsule, septa, and seminiferous tubules inside each lobule, and they are surrounded by loose fibrous connective tissue. And here we can see drawing of histological slide. It's connective tissue capsule or tunica albuginea, which forms septa, which divide tissue of testis into the lobules. Here we can see capsule and septa. Both are formed by dense fibrous connective tissue. And here we have on this picture, we can see sections of seminiferous tubules, transverse and oblique sections. That's why those tubules, they have oval shape, non-round, because this section is oblique. And between them there is loose fibrous connective tissue. It's interstitium of testis or stroma of testis, intralobular stroma, which maintains seminiferous tubules. It contains blood vessels, there are nerves, and also there are Leydig cells which produce hormones. Those small cells which are located between the seminiferous tubules, they produce hormones and testosterone. And these tubules, here we can see numerous cells, and we can find here stems with flagella. So there are spermatozoa and there are numerous progenitors, and it's the site of production of spermatozoa, site of the spermatogenesis. And here we can see tunica albuginea. It's dense fibrous connective tissue or capsule, own capsule of testis. It's mediastinum with arete testis. And here we can see lobuli of the testis, lobules, and they cannot be clearly distinguished from each other on this picture. And here we can see bigger magnification. It's tunica albuginea and numerous sections of convoluted seminiferous tubules, and between them spaces are filled with interstitium, intralobular connective tissue with Leydig cells which produce hormones. And on bigger magnification, we can see on this microphotograph sections of convoluted seminiferous tubules with numerous progenitors of spermatozoa, its site of spermatogenesis, its interstitium, uh, its connective tissue with Leydig cells, and also here we can see it seems like a septa between the lobules. There are different tubules and ducts of the testis which provide main functions of the testis, and they are convoluted seminiferous tubules which are site of production of spermatozoa which provides spermatogenesis. So they also are called tubuli seminiferi contorti or convoluted. Next are tubuli seminiferi recti or straight seminiferous tubules. They are continuation of the convoluted seminiferous tubules, and they carry spermatozoa to the single system of branched system of tubules which is called reproductive. Here we can see reproductive. Rete is network, so network of tubules which collect all spermatozoa from all lobules of the testis, and it's the common site of all spermatozoa of the testis when they are going from the seminiferous tubules. And there are intratesticular system of different ducts. It's seminiferous tract of the testis. And also there are continuations, there are ductuli efferentes which excite from testis and they carry spermatozoa to ductus epididymitis and its beginning of the epididymis. It's another organ which is a part of seminiferous tract. So there are ductuli efferentes which begin in the testis, but they excite from the testis and they carry spermatozoa away from the testis to another following parts of seminiferous tract. And here we can see intralobular connective tissue and also dense fibrous connective tissue which forms capsule and septa of the testis. And here we can see it's tunica vaginalis or continuation of peritoneum which has cavity, it's visceral and parietal layer, and between them there is a cavity which is filled with fluid, and the innermost own membrane or covering of the testis, it's connective tissue capsule or tunica albuginea which forms the septa, and they are going to the mediastinum. And there are seminiferous tubules, convoluted straight seminiferous tubules or tubuli seminiferi recti. It's retrotestis, efferent ductals, and it's ductus epididymitis. It's beginning of seminiferous tract, extratesticular seminiferous tract, and it is continued by ductus deferens. So we'll begin from the main structural and functional units of male reproductive system of the testis. They are convoluted seminiferous tubules. So they are structural and functional units of testis. And they provide main process which takes place in the testis, which is called spermatogenesis, formation of spermatozoa. So it's the site of spermatozoa formation. And these tubules, they are hollow structures, so they have wall, and this wall it has some layers, and they little bit may remind us of structure of another hollow organs, for example organs of digestive or urinary system, but there are some specific features. So the innermost layer of these tubules, it's spermatogenic epithelium. Here we can see this epithelium, and this epithelium is the main layer which provides formation of spermatozoa. So there are epithelial, completely epithelial cells, and also different spermatogenic cells, progenitors of spermatozoa on different stages of their development. It's spermatogenic epithelium, and it's the innermost layer of the convoluted seminiferous tubule. So it's the epithelial specific epithelial lining of the seminiferous tubules. Under them, there is a basal layer, which includes basal membrane and thin layer of connective tissue, fibrous connective tissue. Next layer is myoid layer. It contains cells which are alike smooth myocytes, and in some textbooks you can find that they are exactly the smooth myocytes, but most of histologists they consider them as an connective tissue cells which are very close to myocytes, but they aren't smooth myocytes. That's why they are called myoid cells, which are alike myocytes, but they aren't myocytes. And they have ability for the contraction. They are contractive cells, and they contain actin and myosin filaments. And this contraction of myoid layer uh, results in some kind of peristalsis of seminiferous tubules, and it helps to move spermatozoa through the lumen of seminiferous tubules. And the outermost layer, it's fibrous layer. It's connective tissue, which commonly is described as dense, but this connective tissue is rich of collagen fibers, and it covers seminiferous tubules and protects them, forms them, and its outer fibrous layer, which may be like an adventitia. So it's a spermatogenic epithelium and basal layer, which are alike mucosa, and submucosa myoid layer, which is alike muscular layer, and fibrous layer, which is alike adventitia, but they aren't those layers, so they are specific, and you should know four basic layers of the wall of convoluted seminiferous tubules: spermatogenic epithelium, which has two compartments, basal and adluminal near the lumen, and basal near the basal layer; basal layer, which includes basement membrane and thin layer of fibrous connective tissue; myoid layer, which provides contractions of seminiferous tubules; and the outermost layer is fibrous layer. And here we can see cross section of convoluted seminiferous tubule. The most important layer and the innermost layer, it's spermatogenic epithelium. Here we can see it, and there are different cells, progenitors of spermatozoa. And this pink line, it shows all other layers. They are thin, and they surround this layer of spermatogenic epithelium. Somewhere here we can find basal, myoid, and fibrous layers of the convoluted seminiferous tubules. And here we can see microphotograph, which shows spermatogenic epithelium. Here under spermatogenic epithelium there is a basal layer, under it's myoid layer, and on this picture we can see labeling smooth muscle. That's why some scientists say, some textbooks, they describe, to simplify the description, that myoid layer is muscular layer, but most of histologists, they think that it's myoid but not muscular layer. And the outermost layer is fibrous layer, which covers this muscular layer. So it's the layered structure of the convoluted seminiferous tubule. And here we can see numerous sections, and here we can see seminiferous tubules and its spermatogenic epithelium and all other three layers, which are very thin. And its spermatogenic epithelium and other layers which form the wall of convoluted seminiferous tubules. And now let's go to the innermost and the most important layer of the convoluted seminiferous tubules. It's spermatogenic epithelium, which provides spermatogenesis, formation of spermatozoa. And it consists of two types of cells. There are Sertoli cells or sustentosites, or they may be called nurse cells or shaper cells. Here we can see them. They are completely epithelial cells, and they are resident cells of the spermatogenic epithelium. They form the basis of spermatogenic epithelium. And also there are spermatogenic cells. Here we can see them, numerous spermatogenic cells, which are progenitors of spermatozoa, and they include some following stages. Today we will talk about those stages, but here we should distinguish between Sertoli cells and spermatogenic cells. So spermatogenic cells, they are some kind of transient or alien cells here. They come from the and other structures, and they fill the spaces during the embryogenesis, and they fill the spaces between the Sertoli cells, which are resident cells here. But Sertoli cells, they aren't progenitors of spermatozoa. But what are they doing here? They maintain spermatogenic cells. Here we can see that they have lateral and apical processes. They communicate with each other and they form bases. They keep the spermatogenic cells their so-called arms. They maintain, they keep spermatogenic cells and they form the space for spermatogenic cells. So they form some layers, some compartments where spermatogenic cells are situated, and they form for them special microenvironment. They form them houses where spermatogenic cells rest and develop. So Sertoli cells, they are supportive cells of spermatogenic epithelium, but they are very important in spermatogenesis. And spermatogenic cells, they are coming here. They fill the spaces between Sertoli cells, and they are living here. So they are kept and they are maintained by Sertoli cells, and they are following stages, and they are progenitors of spermatozoa. So here we can see Sertoli cells, epithelial cells with special modified shape, and they maintain spermatogenic cells. And Sertoli cells, they are kind of sustentacular cells. They are anose cells of the testicles, and they help in the process of spermatogenesis when they form microenvironment for spermatozoa. And also they produce androgen binding protein. It's a special protein which provides binding of testosterone, which is produced by Leydig cells. Leydig cells produce testosterone, and Sertoli cells, they produce androgen binding protein. Testosterone cannot make a direct influence on spermatogenic cells. They need special intermediate substance, and its special androgen binding protein, and only complex of androgen binding protein and testosterone can make influence on special spermatogenic cells, so they are sensitive for it. So in simple words, Sertoli cells, they produce special substance which makes spermatogenic cells sensitive to testosterone. And also they provide mechanical support for spermatogenic cells and nutrition for them. Also it uptakes from blood, which surrounds tubules. They uptake from blood different nutrients and give those nutrients for the spermatogenic cells. And also Sertoli cells, they protect spermatogenic cells from different toxins from blood and from autoimmune diseases, because immune system may damage spermatogenic cells. Later we'll talk about this. And Sertoli cells, they form hematotesticular barrier which separates immune system from spermatogenic cells, and Sertoli cells, they play very important role in this formation of this blood-testis barrier. So they are part of blood-testis barrier, and they separate basal and abdominal compartments of spermatogenic epithelium, which is needed to form a blood-testis barrier. And they are activated by follicle stimulating hormone, FSH. This hormone is known as hormone which is regulating functions of female reproductive system. It activates growth of hormones. But this hormone also is present in male organism, and it's produced continuously, as well as luteinizing hormone, LH. So those two hormones, they regulate in females menstrual ovarian cycle, and they have cyclic release, but in male organisms they are produced continuously. Follicle stimulating hormone target cells for the follicle stimulating hormone are Sertoli cells, and target cells for LH are Leydig cells which produce testosterone. So LH and Leydig cells, it's easy to remember, L and L, they luteinizing hormone has target cells Leydig cells, and FSH activates Sertoli cells which are sensitive for this hormone, and under influence of this hormone they produce androgen binding protein which helps to make spermatogenic cells sensitive to testosterone. So let's go to the main cells of spermatogenic epithelium. They are spermatogenic cells. And here we can see Sertoli cells, and on their lateral sides, on their processes, spermatogenic cells are situated. So here we can see cells which are located near the basal membrane. They are called spermatogonia. They are located in the deepest layers of spermatogenic epithelium. The next are primary spermatocytes. Next are secondary spermatocytes. Also there are spermatids, early and late spermatids. And when cell is separated from spermatogenic epithelium, it's called spermatozoon. So there are spermatozoa. And although cells are the following stages, the earliest stage is spermatogonia, later it becomes to be primary spermatocyte, later secondary spermatocyte, later spermatid, and the last final stage is spermatozoon. So they are the following stages, and we can see that the youngest cells are located near the basal layer. So there are spermatogonia. Next are primary spermatocytes, and they are located above spermatogonia. Secondary are located above primary spermatocytes, and spermatids are located near the lumen. So here we can see that during the development of spermatozoa, progenitors of spermatozoa are moving from the basal part of spermatogenic epithelium to the lumen, and there are basal compartment and adluminal compartment of spermatogenic epithelium, and all those stages of the cells, they have special specific localization in spermatogenic epithelium. So let's look how all those cells become to be each other. The first stage, the first cell, which is progenitor of spermatozoa or stem cell of spermatozoa of spermatogenic epithelium, it's spermatogonia. This is a cell which is a diploid cell, and it undergoes some following mitotic divisions. Here we can see division, and it results in the formation of two spermatogonia, and there are some following mitotic divisions of spermatogonia. And after that, spermatogonia, after final mitotic division, it begins to grow, and it increases volume, and it increases volume of the nucleus, and it becomes to be primary spermatocyte. Here we can see one spermatogonia, it becomes to be primary spermatocyte. After that, primary spermatocytes, they enter in first meiotic division. Spermatogonia, they undergo mitotic divisions as usual somatic cells, and primary spermatocytes, they enter in first meiotic division. So first meiotic division, primary spermatocytes. It's easy to remember. They enter in first meiotic division, and result of this division is formation of two secondary spermatocytes. So primary spermatocyte divides into two secondary spermatocytes. It's result of first meiotic division. After that, each secondary spermatocyte enters in the second meiotic division. Secondary spermatocyte enters in second meiotic division. It's also easy to remember. And after this division, each secondary spermatocyte is divided into two cells, which are called spermatids. Here we can see spermatids. So as a result of this, one primary spermatocyte gives rise to two secondary spermatocytes and four spermatids. And after that, spermatids, they undergo some changes. They are round cells, and they haven't flagellum, they haven't tail and acrosome, so they undergo some changes. Special organelles, they appear here, and as a result of this process, spermatozoa develop, and here we can see that final stage of spermatogenesis, it's formation of spermatozoa. So the following stages of spermatogenic cells are spermatogonia, primary, secondary spermatocytes, spermatids, and spermatozoa. And there are some changes in the chromosomic number in the karyotype of all those cells, because spermatozoa, they are haploid cells, they are gametes, and they are necessary for the fertilization. So they contain only half of genetic material, and spermatogonia, they are similar in their karyotype to all somatic cells. So let's look how it's provided. And first of all, let's look at the stages of spermatogenesis. So first stage of spermatogenesis, it's called proliferation or mitosis. Second, it's spermatocytogenesis. It results in formation of spermatocytes. Then spermatogenesis, which results in the formation of spermatids, and the last stage is spermiogenesis, formation of spermatozoa. So there are three main stages: spermatocytogenesis, spermatidogenesis, and spermiogenesis. And before them there is special preliminary stage which is called proliferation or mitotic divisions of spermatogonia. And those stages you should remember, and also you should remember what are the processes which take place during those stages and what are the results of those stages. So first stage is proliferation of spermatogonia. Here we can see them, and it may be considered as a preliminary stage of spermatogenesis. So they are diploid cells which are like all somatic cells. Their karyotype is 2N, so they have 2 chromosomes in pair and 4C. It's because of and 2C because of amount of DNA. And spermatogonia, they undergo some following mitotic divisions. There are type A dark spermatogonia, which are stem cells, and type A light spermatogonia, and type B. So type A and type B. Type A is divided into dark and light. Type A dark cells, they aren't active, and light type A and type B spermatogonia, they are active, and they divide rapidly by mitosis, and they form new generations of cells. So here we can see type A spermatogonia and type B. Type A dark cells, they are stem cells, they are inactive, and only after injury of another cells they may be activated to provide regeneration of another spermatogenic cells. Type A light cells, they are more active than dark cells, but main cells which provide proliferation, they are type B spermatogonia. And there are some following mitotic divisions, and we can see that their number increases. And there are numerous cells of which are result of divisions of type B spermatogonia, and they have the same genetic material. They have the same karyotype, so it's simply divisions of type B spermatogonia, alike divisions of all somatic cells. And spermatogonia during their divisions, specific feature of all spermatogenic cells is they save cytoplasmic bridges. Here we can see it. It's first division of spermatogonia, second mitotic division of spermatogonia, and there are cytoplasmic bridges between the cells, and they form syncytium. It's special term which describes supracellular structure, which includes some cells which are connected by cytoplasmic bridges. It's incomplete divisions of cytoplasm. So spermatogonia and all following stages except spermatozoa, they have cytoplasmic bridges between each other. And final division of type B spermatogonia, it results in the formation of primary spermatocytes. Here we can see that some cells, they excite from this mitotic cycle, from following numerous mitotic divisions, and they begin to grow. And they undergo also reduplication of DNA. So from 2N to C, they become to be 2N4C. Their nucleus increases in size. The cell passes through the synthetic period and increases in size, and also they become to be primary spermatocytes. So here we can see the difference in the appearance of the type B spermatogonia and primary spermatocyte. It's bigger and it has bigger nucleus because it has passed through the reduplication of DNA. So it's primary spermatocyte, which is the second stage after the spermatogonia. And now let's look what will happen with primary spermatocyte. Here we can see spermatogonia, and they form primary spermatocytes, big cells, the biggest cells in the spermatogenic epithelium, and they have undergone reduplication of DNA. And primary spermatocytes, these big cells, they enter to meiosis, and first it enter to interphase before division, grow in size, and appear larger than spermatogonia. Here you can see this size. and they can enter the prophase of the first meiotic division, which is extremely prolonged, about 22 days. So here we can see how long is this process. That's why reproductive function needs to reduce different toxic substances, different alcohol drinks when a couple is planning to have a baby, because spermatogenic period is very long, and during about 3 months pair couple shouldn't apply different drugs, different toxic substances, alcohol, because it may cause different mutations in the spermatogenic cells. So they enter in the prophase of the first meiotic division, and after this it's continued, and first meiotic division results in the formation of 2 secondary spermatocytes. From one, there are 2 secondary spermatocytes. So the completion of the first meiotic division results in the formation of 2 secondary spermatocytes, and we can see them. It's called spermatocytogenesis, formation of primary and secondary spermatocytes. And next stage, it's spermatidogenesis. Secondary spermatocytes, they enter and complete the second meiotic division. And their division results in the formation of 4 spermatids, which lie in the luminal part of the seminiferous epithelium. So 4 small cells, they are formed from 2 secondary spermatocytes, which in on order were formed from 1 primary spermatocyte. So here we can see 2 secondary spermatocytes and 4 spermatids, which are a result of second meiotic division. So 1 primary spermatocyte gives rise to 2 secondary spermatocytes, and they give rise to 4 spermatids. It's a result of second meiotic division, and it's a result of spermatidogenesis. And here we can see spermatogonia, primary, secondary spermatocytes, and spermatids. And after that, spermatids, they undergo some changes and they become to be spermatozoa. So there are 2 meiotic divisions. First meiotic division is division of primary spermatocyte, and second meiotic division is division of secondary spermatocytes. First division is reductional, and second division is equational. So what does it mean? Here we can see, let's look at the karyotypes of different cells and what changes to occur during spermatogenesis. So spermatogonia, they have karyotype 2N, 2C. 2N. Here we can see 1 pair of chromosomes. There are 23 pairs, but we'll look at 1 pair and 1 chromosome and another chromosome. Both chromosomes are made up of 1 chromatid. It's appearance of chromosomes before the duplication of DNA. All somatic cells, before they pass through the synthetic period of interphase, they have this karyotype 2N, 2C. And spermatogonia, they become to be primary spermatocytes. They grow, and on early stage they have primary spermatocytes karyotype 2N, 2C, but they pass through the reduplication of DNA, and each chromosome from 1 chromatidic becomes to be double chromatidic, X-like appearance, and number of copies of DNA in each pair becomes to be 4. From 2C karyotype becomes to be 4C. 1, 2, 3, and 4. 4 chromatids in pair are present, and that's why karyotype is 2N, 2 chromosomes, but 4C, 4 chromatids. It's a karyotype of primary spermatocytes. And primary spermatocytes, they enter in first meiotic division, and 2 secondary spermatocytes are developed. And you should know that feature of first meiotic division, that not chromatids, chromosomes are divided and they are moving to different cells. So secondary spermatocytes, they have 1 chromosome from pair, 1N, but each chromosome is double chromatidic. 2C, 1 and 2 chromatids, but 1 chromosome, 1N, 2C. It's karyotype of secondary spermatocytes. And And second meiotic division, when secondary spermatocytes are divided into four spermatids. And in this division, second meiotic division, it reminds of typical mitotic division, and chromatids are going to the daughter cells, and here we can see that chromatids are divided between the cells, and each cell has one N, it's a haploid set of chromosomes, one N, but one C. You can see here one C. So it's spermatids, it's karyotype of spermatids. And spermatozoa, they also have this karyotype 1 and 1C, because only changes in the structure and appearance occur when spermatids they become to be spermatozoa. So here we can see stages and features of the karyotypes of different spermatogenic cells, from typical somatic cells to N2C cells become to be haploid cells 1 and 1C, and it's a result of spermatogenesis. So here we can see spermatogonia 2 and 2C. Mitotic divisions, they don't include changes in karyotype. As a result, primary spermatocytes 2 and 4C, secondary 1 and 2C, spermatids 1 and 1C, and spermatozoa as well 1 and 1C. So here we can see changes in karyotypes of all those cells. And here we can see result of spermatogenesis. Four spermatids are developed here, and they have haploid set of chromosomes 1 and 1C. And after that, they change their shape. Flagellum appears here, and they become to be alike mature spermatozoa, but till the moment when they are separated from Sertoli cells, they still are called spermatids. And only at the moment when cell is separated from the Sertoli cell, it's called spermatozoon. So spermatozoa, those cells may be called spermatozoa only after the separation from Sertoli cell. And next stage after the spermatogenesis, it's spermiogenesis or formation. So what is spermiogenesis? It's development of mature spermatozoa. Here we can see that cells, spermatids, they have round shape, they have simply shape and structure, and after that they become to be cells with specific shape, which helps to provide their movement, to provide fertilization, and all those changes, they occur during the last stage of spermatogenesis. It's called spermiogenesis. Spermatids are small round haploid cells, and terminal phase of spermatogenesis is called spermiogenesis and consists of the differentiation of two of the newly formed spermatids into spermatozoa. And here we can see this process of transformation. What happens during this process? Here we can see that first of all, a chromosome develops. A chromosome, it's organelle, special organelle which develops from Golgi apparatus, from Golgi body, and it's special single membranous organelle which contains hydrolytic enzymes which are needed to provide fertilization. It dissolves membranes of the ovum. And flagellum occurs. It develops from cytoplasm, and later special microtubules, they form the axoneme of the flagellum. Its tail, developing tail of spermatozoon, and excess of cytoplasm is separated from the head of spermatozoon, and it forms residual bodies. They are separated from the head of spermatozoon, and also mitochondria, they are moving to the mid piece of spermatozoon from the head, and here in this region there is a proximal centriole and basal body of the axoneme of the tail. It's known as a distal centriole, which doesn't enter during the fertilization in cytoplasm of ovum. So as a result of spermiogenesis, there is head, midpiece, and tail of spermatozoon, and between them there is a neck in this region. A chromosome helps to dissolve membranes of the ovum. It contains hydrolytic enzymes. Also, there is haploid nucleus without nucleolus, and spermatozoon is the only cell which doesn't have the nucleolus, and it has haploid set of chromosomes. And also, proximal centriole is present here, which enters in the cytoplasm of ovum during the fertilization as well as nucleus. So zygote receives centriole from father and mitochondria from mother. Mitochondria, they are located in midpiece. They produce energy to provide mobility of the tail, but they don't enter in cytoplasm of ovum. That's why all mitochondria of zygote of children are received from mother's cell. And here we can see, let's look once again, and let's repeat the stages of spermatogenesis. Here we can see maternal cell of all spermatogenic cells. It's spermatogonia, and numerous spermatogonia, especially type B, they have numerous mitotic divisions, and their number increases. Its proliferation. After that, it increases number of chromosomes, and nucleus grows, and also entire cell grows, and it becomes to be primary spermatocyte. Primary spermatocytes, they enter in first division of meiosis, and it results in the formation of two secondary spermatocytes. Here we can see them. Secondary spermatocyte enters in the second division of meiosis, and it results in the formation of spermatids. Here we can see them. Spermatids, they enter in the spermiogenesis. They change their appearance. Special organelles are developed, and they become to be spermatozoa. And here we can see final stage of the spermatogenesis, formation of spermatozoa. But you should remember that late spermatids, they haven't any changes in their appearance with spermatozoa. It's early, it's late spermatid, but we can call this cell spermatozoon only after the separation from Sertoli cell. And the stages of spermatogenesis are the following. First, it's preliminary stage, proliferation or mitosis. Next, spermatocytogenesis. Next, it's spermatidogenesis, and the last stage is spermiogenesis. It's classification of the stages of spermatogenesis, which is accepted, and it's the common classification in the English textbooks, but in Ukrainian, Russian textbooks there is another classification which is accepted, and there are proliferation, growth, maturation, and formation. 4 stages instead of 3. In this classification, proliferation is preliminary stage, and there are 3 main stages. Spermatocytogenesis, which results in the formation of primary and secondary spermatocytes. Spermatidogenesis, which results in the formation of spermatids. And spermiogenesis, which results in the formation of spermatozoa. 3 stages. And these correspond, together with preliminary stage, to the stages in Ukrainian textbooks. First stage is proliferation, mitotic divisions of spermatogonia. Second stage is growth, when spermatogonia are growing and they become to be primary spermatocytes. This stage corresponds to spermatocytogenesis. Next stage is maturation. It includes 2 meiotic divisions, and it results in the formation of 4 spermatids. It corresponds to spermatidogenesis. And the last stage is formation, or spermiogenesis, formation of spermatozoa. So it's another classification which includes 4 stages of spermatogenesis. This classification is accepted in Ukrainian textbooks, and this is accepted in different Western textbooks, in English mainly. So different classifications, but they correspond to each other. And here we can see this spermatogonia. They are located in the basal pole in the basal compartment of the spermatogenic epithelium. They are located near the basal membrane. Next stage, it's primary spermatocyte, and spermatogonia after growth, it migrates in the upper part of the spermatogenic epithelium. So during their development, they aren't stand on the same place. They are moving to the lumen. And primary spermatocyte is located above the spermatogonia. It goes upper. After that, primary spermatocyte gives rise to two secondary spermatocytes, which also are going to the lumen. Here you can see primary, and it becomes to be two secondary spermatocytes, and they are going upper. And each of secondary spermatocyte gives rise to spermatids. And here we can see formation of four spermatids from two secondary spermatocytes, and they are located near the lumen. Here we can see these spermatids. So during all the stages of spermatogenesis, cells are moving from the basal part of spermatogenic epithelium to the lumen. It's a luminal compartment of spermatogenic epithelium. And after that, spermatids, they change their shape. They become to be late spermatids, and after that they are separated and they are called spermatozoa. And here we can see formation of spermatozoa. They are located in the lumen. Here we can see spermatogonia, primary, secondary spermatocytes, spermatogonia early, late, and spermatozoons are located in the lumen. And here we can see microphotograph of convoluted seminiferous tubules. Here we can see section. Inside the lumen we can see some cells with tails, and near the basal pole there are different cells. The smallest cells near the basal pole there are spermatogonia. Bigger cells, the biggest, are primary spermatocytes. Near the lumen there are secondary spermatocytes and spermatids. As spermatozoa, they are located in the lumen. Here we can see them, and they are going to be separated from Sertoli cells. And here we can see these cells under microscope. It's drawing histological slide. Spermatogonia are near the basal pole. Primary spermatocytes, the biggest cells with big nuclei, secondary spermatocytes, and spermatids. And we can see here that spermatids in different tubules are on different stages. We can see here spermatids which are completely round. They are early spermatids. Spermatids with short tails, and with long tails which are ready to be separated and to become to be spermatozoa. So in different seminiferous tubules, spermatogenesis is on different stages. Some tubules, they are on the final stages of spermatogenesis, some are on the early stages, some are on the intermediate stages, and it keeps continuous presence of mature spermatozoa in the spermatogenic tubules in the testes, and it provides continuous fertility. And also you should know about a special structure, special phenomenon in the testes, which is called blood-testis barrier. And there are diploid spermatogenic cells and haploid spermatogenic cells. You know that spermatogenic cells during spermatogenesis, they become to be haploid from diploid. And diploid spermatogenic cells are located in the basal compartment. There are spermatogonia and primary spermatocytes. They are diploid. They have karyotype 2N. And haploid spermatogenic cells are located in the abdominal compartment. There are secondary spermatocytes and spermatids. They have a haploid set of chromosomes. Here we can see that spermatogonia 2N, 2C. Primary spermatocytes on first stage they are 2N, 2C, and after that they are 2N, 4C. And beginning from secondary spermatocytes until spermatozoa, they are haploid. So, what is the reason to know this? Very important thing that immune system checks karyotype of the cells and decides is the cell own or foreign. And when immune system meets this cell 2N, 2C, it thinks that this cell is own and doesn't damage this. Primary spermatocytes on stage 2N, 2C also are own for the immune system. But when primary spermatocytes enter in prophase, they begin crossover of the chromosomes, chromatids, and especially secondary spermatocytes, spermatids, and spermatozoa, which are haploid, they may be found by immune system, and immune system will kill them because it thinks that they are foreign. And because it checks their karyotype and it may kill them. So how to prevent this? and there is a blood-testis barrier. It separates spermatogenic cells and cells of immune system. And there are basal and abdominal compartments. Basal compartment, it contains diploid cells, which shouldn't be separated from immune system. They are located near the base of membrane, and they are diploid and immune system. Some cells of immune system which provide immune supervision, they are present in the capillaries, and they describe them as own. But in other cells, haploid and primary spermatocytes which are entering the prophase, they should be separated from immune system. They should be enclosed in the spermatogenic epithelium, and it's provided by blood-testis barrier. It prevents autoimmune reactions because immune system may damage own spermatogenic cells. So abdominal compartment includes haploid spermatogenic cells and primary spermatocytes in the process of the meiosis when they have entered in the meiosis to prevent autoimmune reactions and to prevent killing of own spermatogenic cells by immune system. And what are the structures which provide blood-testis barrier? First of all, there are Sertoli cells and their lateral projections. They form the barriers and they isolate basal compartment from the abdominal. And here we can see this intermediate part where primary spermatocytes are separated both from spermatogonia and from secondary spermatocytes. So cells from this compartment, they pass through this first barrier, they enter here, and after this they are entering in the abdominal compartment. And Sertoli cells, they are the most important part of blood-testis barrier. Also, it includes layers of the wall of the convoluted seminiferous tubules, so it's basal, myoid, and fibrous layer, and wall of the capillary, which includes basal membrane and endothelium, endotheliosite. So there are layers of blood-testis barrier, and it prevents autoimmune reactions. Those autoimmune reactions, they may occur especially in viral parotitis. It's viral disease which causes inflammation of parotid gland, but also this virus may cause in boys, especially in young boys, inflammation of the testis. It's called orchitis, and in some cases, blood-testis barrier is broken and immune system goes to the spermatogenic epithelium and kills own spermatogenic cells, and often it results in the male infertility. So blood-testis barrier is very important to keep spermatogenic cells safe. And here we can see layers of the blood-testis barrier. There are Sertoli cells and their lateral projections. They form tight junctions, and there are spermatogonia. They are located in the basal compartment. It's wall of spermatogenic convoluted seminiferous tubule and wall of the capillary endothelium and basement membrane. And there are the layers of the blood-testis barrier, Sertoli cells, wall of the convoluted seminiferous tubule, and wall of the capillary. And here we can see also Sertoli cells, and they form this blood-testis barrier. They are the most important cells which form this barrier. And also another function of testis, it's endocrine function, and it's provided by Leydig cells. Here we can see part of the convoluted seminiferous tubule with different cells on different stages of their development, spermatogenic cells. And between the tubules, there are inclusions of the special endocrine cells. They are called Leydig cells. They are cells of connective tissue, interstitial connective tissue, loose fibrous connective tissue, which lies between the convoluted seminiferous tubules. But there are special endocrine cells which produce testosterone, main male hormone. And they are controlled by LH, luteinizing hormone, which is known as hormone of female reproductive system, but also it's produced in male reproductive system continuously, and this hormone has target cells. They are Leydig cells which produce testosterone. And here we can see Leydig cells. They have acidophilic cytoplasm because of good development of smooth endoplasmic reticulum, because they produce steroids. Androgen is steroid, and it has a lipid structure, and they produce this hormone, and they are located between the convoluted seminiferous tubules. And here we can see convoluted seminiferous tubules, and between them pink small cells are Leydig cells. They form groups, and those cells they produce hormones. And here we can find groups of Leydig cells. They are main endocrine cells of the testes. And here we can see convoluted seminiferous tubules and group of Leydig cells, which is located in the interstitium stroma inside the lobule. Here we can see Leydig cells which produce testosterone. Main structures of the testis are lobules, which include convoluted seminiferous tubules and also interstitium, those fibrous connective tissue which fills the spaces between the tubules. And also there is a seminiferous tract, which includes tubuli recti, straight tubules which begin directly from the convoluted seminiferous tubules, and they are alike ducts which excite from the lobules, and those tubuli recti or straight tubules, they are opened in the retrotestis, special network here. After that, spermatozoa are moving to the ductuli efferentes, which carry spermatozoa to the ductus epididymitis. It's formed by a special duct. It's coiled, and after it, it's continued by ductus deferens, which is opened into ductus ejaculatorius, and it's opened in urethra, which carries spermatozoa to outer environment. And let's talk about them step by step. So intratesticular parts of seminiferous tract are tubuli recti, retrotestis, and initial portions of the ductuli efferentes. And here we can see ductuli recti, retrotestis, and ductuli efferentes, and its epididymis, which is formed by ductus epididymitis, and it's continued by ductus deferens, and it's fused with ductus, this duct from the seminal vesicles, and they form ductus ejaculatorius, which is opened in urethra. So organs and parts of the seminiferous tract, they are hollow structures. They have lumen and they have wall, and this wall is composed of 3 layers. They are mucosa, muscularis, and adventitia. Mucosa is composed of epithelium, and epithelium may be different in different parts of seminiferous tract. And under epithelium there is space membrane and loose fibrous connective tissue or lamina propria, and it's usually very thin. Muscularis, layer of smooth myocytes, and the outermost layer is adventitia, also loose fibrous connective tissue, which usually is continued into the surrounding tissues. And here we can see epithelium of mucosa, muscularis, layer of pink structures, and the outermost layer more light. It's adventitia. Ductuli recti and retrotestis, they are lined with this type of epithelium, simple or cuboid, or in some the thinnest tubules it may be cuboid or squamous. The thinnest tubules in the retrotestis, they have simple squamous epithelium. Tubuli recti, they have columnar or cuboid epithelium. So it depends on diameter of the tubules. Tubuli recti columnar epithelium, and retrotestis cuboid or even squamous in the smallest tubules. And it's continued by the ductuli efferentes. Here we can see they carry spermatozoa from the testis to the epididymis, and it's composed of ductuli efferentes. They are included in the head of epididymis, and also it has body and tail, and there are ductuli efferentes which connect testes. They have intratesticular part and extratesticular part which forms the head of epididymis, and they enter in the same in the single duct, which is called ductus epididymitis, and it's highly coiled and is continued into the vas deferens. So what are the ductuli efferentes and their histological structure? They have wavy contour because of different cells. There are cells with cilia, ciliated cells, they are tall, and shorter cells which are secretory cells, which produce special fluid which helps in the movement of spermatozoa. That's why group of tall cells is changed by group of short cells, and it results in the wavy contour of the ductuli efferentes. And here we can see specific histological feature of ductuli efferentes. It's wavy contour. And under them we have on this picture it's muscularis and adventitia. So we can see here three basic layers of the wall of ductuli efferentes. And in the lumen there are spermatozoa which are moving from testis to the vas deferens. And ductuli efferentes, they are opened into ductus epididymitis, and it has also in the lumen spermatozoa. It's bigger in diameter, and we can see that this epithelium is pseudostratified. There are some rows of the cells, and it's ciliated. So ciliated cells and epithelium is pseudostratified, but it's not stratified because all cells, they have contact to base membrane. And it's not stratified. And also muscularis is present here, and adventitia is continued into surrounding connective tissue. So this histological slide, it's called epididymis. It's section of epididymis. It may contain both ductuli efferentes, which form the head of the epididymis, and they are opened in the ductus epididymitis, and we can distinguish them by the epithelium in ductuli efferentes. The contour is wavy because of two kinds of cells, and in ductus epididymitis all cells are tall, superficial cells are tall, and contour is smooth. And both epithelia, they have cilia which help in the movement of spermatozoa. And on this picture we can see ductuli efferentes which connect testis to the ductus epididymitis, and here we can see this ductus with smooth contour, and between them there is connective tissue stroma, loose fibrous connective tissue, which fills the spaces between those ducts. And ductus epididymitis is continued into vas deferens or ductus deferens. Ductus deferens is similar to ductus epididymitis, but there are some features. Epithelium is pseudostratified ciliated columnar as well as in ductus epididymitis. And it forms mucosa, its epithelium and loose fibrous connective tissue. Under it there is a muscularis, and muscularis, it has three layers, and it's very well developed because vas deferens is muscular organ, and contractions of muscularis, they act like a pump. They help in the movement of spermatozoa. So there are three layers, inner and outer are longitudinal, and middle layer is circular. So three layers and work, and they provide peristalsis of vas deferens. And the outermost layer is adventitia, which covers muscular layer. And here we can see vas deferens with cilia or stereocilia, which help in the movement of spermatozoa. It's pseudostratified columnar epithelium, and there are basal cells. They are located near the base membrane. They provide regeneration. And under it there is lamina propria, loose fibrous connective tissue, which underlies the epithelium and provides it its nutrition. And here we can see testis. There are ductuli efferentes which are opened in ductus epididymitis, and it's continued into vas deferens, which goes through the inguinal canal, and after that it goes to the urethra. And here we have on this picture it's seminal vesicle. We can see here it and the seminal vesicle, it produces fluid. It's first accessory gland, and duct of the seminal vesicle and duct of the ductus deferens, they are fused, and they form ductus ejaculatorius, which is opened into the male urethra. Here you can see urinary bladder and its urethra, its prostatic part of urethra, and this duct is opened here. Sperms, they are coming here, and fluid from the seminal vesicles also comes here. It's ductus ejaculatorius. And there are accessory glands. It's a seminal vesicle, two pairs, seminal vesicles, and ductus deferens. They are fused, they form ductus ejaculatorius, which is opened into urethra. Also, there is prostate gland, and also there are bulbourethral glands. Prostate, seminal vesicles, and bulbourethral glands, they produce different kinds of fluid which forms environment for the spermatozoa. And spermatozoa, they are coming here. They also have some fluid which is produced by the epithelium of the seminiferous tract, and additional fluid is produced by accessory glands. And in urethra, all those components are mixed, and final content of semen is produced. And after that, it moves through the urethra to the external environment. And male urethra, it's the last part of the seminiferous tract. It's organ of two systems, of urinary system, it's way for urine, and also it's organ of reproductive system, where spermatozoa are going to the outer environment. So male urethra, it has four layers. It's mucosa with transitional epithelium in initial portions, and it transits into the stratified squamous non-keratinized epithelium. Submucosa, muscularis, and adventitia, four layers which are similar to the organs of urinary system. Anatomical parts of male urethra are preprostatic part of urethra, it's above prostate gland, prostatic urethra, where urethra passes through the prostate gland, membranous urethra, where urethra passes through the muscles of perineum, and spongy urethra when it passes through the spongy bodies. So there are anatomical parts of urethra, and they have features in the epithelial lining. Preprostatic and prostatic urethra, they have transitional epithelium, which is characteristic for the urinary system. And after that, there are some transformation of epithelium of male urethra. In membranous part, epithelium is simple columnar or pseudostratified. In spongy urethra, epithelium, first of all, is simple columnar epithelium, then it becomes to be simple columnar pseudostratified. After that, it becomes to be stratified squamous non-keratinized, and after that, it transits into keratinized epithelium. In distal part, it transits into the epidermis of skin. So here we can see transition from transitional epithelium into epidermis of skin. And those transitions, they are present in different parts of urethra. So here we can say it's lumen of urethra and its mucosa, and it's surrounded by a spongy body in spongy part, and it's rich of venous vessels, its cavernous tissue, venous blood vessels. They are muscular veins or veins with poor development of muscles. They are filled with blood, and between them there are layers of loose and dense fibrous connective tissue which keep them in open condition. And now we are going to the accessory glands which produce fluid which is necessary for the movement of spermatozoa. There are three groups of glands. There are seminal vesicles, it's prostate, and bulbourethral glands. Seminal vesicles, they form sperm coagulum, and which helps in the regulation of sperm motility. They produce a fluid which is rich with fructose, which is necessary for the nutrition of spermatozoa, and it's needed for their movement. And their ducts are opened, they are fused with ductus deferens, and they form ductus ejaculatorius, which is opened in urethra. Prostate, it produces fluid which is rich with mucus, and also it liquidifies the semen. And bulbourethral glands, they produce mucus. Also, it's necessary to move spermatozoa. So first glands are seminal vesicles or vesicula seminalis, and they form sperm coagulum and regulation of sperm motility, and also this secret, it provides the suppression of immune function in the female genital tract, because immune system of female organism may break down spermatozoa, and it may cause female infertility because of immune conflict between female organism and spermatozoa. And these seminal vesicles, they produce special substances which decrease immune function in female reproductive tract. Here we can see seminal vesicles. Their ducts are opened in urethra, and it helps in the movement of spermatozoa. Histological structure of the seminal vesicles, they are hollow organs, and they are alike this duct vas deferens. So they have adventitia muscularis and mucosa, and lining of mucosa is or simple columnar or simple pseudostratified epithelium, and this epithelium is secretory epithelium which forms this fluid. Here we can see pseudostratified columnar epithelium which lines those vesicles. It's epithelium, it's mucosa. Under it there is muscularis which helps to push out those secret. And the outermost layer, it's adventitia, it's simply loose fibrous connective tissue which covers the seminal vesicles. Next gland, it's prostate gland. Here we can see, and prostate gland is specific gland which has some specific functions. It's at the same time exocrine, endocrine gland, and also muscular sphincter. So let's look at the first function. It's exocrine function, and prostate gland is rich of different exocrine glands which produce mucus, which produce fluid, and they are opened, secretory units are opened in the ducts which are opened into urethra, and this is necessary to move spermatozoa. So the secret of prostate gland is released in urethra. Prostate gland, it contains 30 or 50 tubular alveolar glands, which are empty into 15-25 independent excretory ducts, and they are opened into the urethra. There are three types or three groups of prostatic glands, and here we can see three regions which are located near the urethra. It's urethra, it's lumen of urethra, and its wall of urethra. The peripheral region of prostate, it contains main glands, so-called main glands. They are the largest, and their ducts run posteriorly, and they are opened into urethra. So there are main glands of the prostate gland, and they have the biggest volume. Here we can see the second zone, more internal zone. They are located near mucosa of urethra, so this zone is called submucosal, and glands are also called submucosal glands. They also are opened into big ducts which are opened into urethra. And the innermost zone under mucosa of urethra, it's called mucosal zone, and glands are called mucosal. These glands are mucosal, these are submucosal, and those glands are main or chief glands. So these glands, the smallest in the innermost zone, they have distinct ducts, and they are opened directly to the urethra. So you should know that there are three zones. It's mucosal, submucosal, and peripheral zone, and three groups of glands of prostate: mucosal, submucosal, and main or chief glands of prostate gland. There are the biggest glands are main or chief, and they produce the biggest amount of product of prostate gland. So it's exocrine function of prostate gland. But it's a mixed gland, and prostate gland, it also has endocrine function, and it produces prostaglandins. That's why they were called prostaglandins, because they were found for the first time in the prostate. That's why they are called prostaglandins. And it's a huge group of different hormones, prostaglandins, and some of them they participate in the inflammation, some of them they regulate blood pressure, some of them they regulate functioning of the gastric mucosa, and those which are produced in prostate gland, they also are involved in the regulation of function of male reproductive system. So it's endocrine function of prostate gland. So it has exocrine function, endocrine function, two functions of prostate as a gland, but also prostate is a muscular organ. Here we can see glands, exocrine glands. Between them there is connective tissue stroma. Parenchyma is composed of glands. Stroma is connective tissue between them, and also there are numerous smooth myocytes which are located in the stroma, and that's why prostate acts like a sphincter. It is able for the constrictions, and it's located near the neck of the urinary bladder. It surrounds urethra, and its additional sphincter of male urethra. So here we can see that prostate gland, it has fibromuscular stroma. There are smooth muscles and connective tissue, and this connective tissue is rich in collagenous and elastic fibers. So it forms fibromuscular stroma of the gland. And glands, they are have an cuboidal or columnar epithelium in glands and in the ducts. So it's exocrine or parenchymal part of the prostate gland. And let's look at histological slide. It's drawing of microphotograph of prostate gland. It's urethra. It's lined with transitional epithelium. There are mucosal glands which are opened directly in urethra. There are submucosal glands and peripheral glands, or chief and main glands, and they form groups or lobules, and it's fibromuscular stroma between the glands. There are secretory units of the glands. They are branched, and the epithelium is columnar or cuboidal. And also in aging, there are some concrements, stone-like inclusions inside the lumens of secretory units of prostate gland. It's sign of aging. It's a normal condition, and those inclusions are present in aged persons. So it's a structure of prostate gland. It's capsule, septa, and fibromuscular stroma, which acts like a sphincter. So here we can see structure of the prostate gland. It has three functions: exocrine, endocrine, and also contractive or sphincter function. And the last glands of male reproductive system are bulbourethral glands. They are mucous glands which are like salivary mucous glands, and they produce special mucus which is released in the ducts which are opened in the urethra. And also secret of bulbourethral glands helps in the movement of spermatozoa. So let's make a conclusion how organs of male reproductive system are related. Here we can see convoluted seminiferous tubule. It has a spermatogenic epithelium, basal, myoid, and fibrous layers, and they are lumen, and spermatozoa are formed here, and also some fluid is produced, especially by Sertoli cells and in other cells of the following parts of seminiferous tract. And spermatozoa are going to the seminiferous tract. Epithelium of seminiferous tract produces some fluid, and also contractions of muscles of myoid layer and muscular layer in the seminiferous tract. It helps to move spermatozoa to the place of destination. And also glands, accessory glands, they produce fluid, special fluid, which is needed to form the semen. And there are two components. It's fluid, which contains nutrients which are needed for the movement of spermatozoa, and also special mucus in other products, water, which provide and increase sperm motility. And the main cells and main structures in the semen are spermatozoa, which are moving through the seminiferous tract to the outer environment. And the very last stage of their development takes place in the female reproductive tract. It's called capacitation, when some parts of the glycocalyx are removed, and spermatozoa are activated, and they are able to penetrate membranes of the ovum. So the very last stage of their development takes place during the movement via female reproductive tract. And here we can see that sperms and fluid goes to the outer environment. And here we can see how the organs and parts of the male reproductive system are related. Uh, it's the testis which provides production of spermatozoa and also hormones. Spermatozoa are going to the epididymis, from the epididymis to ductus deferens, from ductus deferens to ductus ejaculatorius, which also receives fluid from seminal vesicles. All those substances are going to urethra. Also, products from prostate gland are released to urethra, as well as product of bulbourethral glands. And final mix of fluids and spermatozoa is going to the spongy urethra and to outer environment. So here you can see relationships and microscopic structure of the organs of male reproductive system. So students, that's all, and thank you for your attention.